Carbohydrates are really central to plant physiology, so we're going to start out with talking about the three main categories of carbohydrates. Carbohydrates are all contain carbon, hydrogen, and oxygen. So sometimes I'll abbreviate them by writing CH2O. They often have other elements in them as well, such as phosphorus or nitrogen, but they all generally have this empirical formula CH2ON. So some repeating combination of carbon, hydrogen, and oxygen. And I'll talk about three main categories of carbohydrates. So there's monosaccharides. Those are the simple sugars. There's oligosaccharides, and those are essentially two simple sugars bonded together. So a molecule containing two simple sugars bonded together. There may occasionally be three, but generally it's two simple sugars. And then the polysaccharides are polymers consisting of many, so hundreds or thousands of simple sugars bonded together. So we'll talk about examples of these three categories and their functions in tree physiology. The first category is monosaccharides. So monosaccharides are just simple sugars and their derivatives. They're the basic carbohydrates from which the more complicated oligosaccharides and polysaccharides are derived. So they are built out of the monosaccharides. Here are two examples of some of the most common monosaccharides, glucose and fructose. These are both 6-carbon sugars. Glucose is the primary product of the Calvin cycle. So it's the primary sugar that is derived from photosynthesis. It's also the primary sugar that's metabolized in respiration. Fructose, as its name implies, is commonly found in fruits. Phosphorylation of monosaccharides is usually required for sugar transformation reactions. So when there's a chemical reaction in which one type of sugar is transformed into another type of sugar, that usually involves the phosphorylation of that sugar. So for instance, if we have sugar A, that sugar A will be phosphorylated. It'll have a phosphate group added onto it. You can see that because that requires ATP, that's an energy requiring step. So the expenditure of energy is required. And then once we have that phosphorylated sugar, sugar A in that case, that can be involved in another energy requiring reaction where that sugar A is then transformed into sugar B. So that's a transformation reaction. We've already talked about a sugar transformation reaction, but I didn't really elaborate much on that phosphorylation. But recall that RUBP, well, first, maybe backing up, what reaction is this? This is the first reaction in the carbon cycle, in the Calvin cycle. So note that this reaction is catalyzed by rubisco. And this is the carboxylation, the adding of a carbon molecule or a carbon atom onto RUBP. And recall, RUBP, I defined that previously as a 5-carbon phosphorylated sugar. Okay, so this is an example of a sugar transformation reaction where that 5-carbon RUBP, ribulose bisphosphate, that phosphorylated sugar is transformed into some other form. So this is an example of a phosphorylated sugar transformation that we've already talked about in this class. The next category of carbohydrates is oligosaccharides. So like I mentioned before, oligosaccharides is the linkage of two or a few is what I usually call it. Might be two, maybe sometimes three monosaccharides linked together. This example, both examples I'll show you is just two monosaccharides linked together. In this case, we have a glucose molecule bonded onto a fructose molecule. So you can see these are linked together. So this larger molecule is sucrose. So sucrose is a glucose and a fructose monosaccharide bonded together. Sucrose is the most important oligosaccharide in plants because it is the primary sugar transport molecule. So when sugars are transported in the phloem, they are usually transported in the form of sucrose. Sucrose, the reason that this is a primary sugar transport molecule, or the reason that sugars are transported this way, is that this form is less reactive. So if glucose or fructose, simple sugars, were transported, those easily react biochemically, and the formation of sucrose essentially makes a more stable and less reactive sugar form that can be transported to a final destination where it is broken up again, and then glucose or fructose can be metabolized at the sink. Sucrose is table sugar. So when you use sugar in your coffee or in a recipe, you're using sucrose, often derived from phloem of, for instance, sugarcane. Here's another example of an oligosaccharide. This is maltose. Maltose is just two glucose molecules bonded together. So just another example of an oligosaccharide. The third category of carbohydrates is polysaccharides. Polysaccharides are polymers. Polymers are just very long molecules consisting of many simple units repeated many times and linked together into a larger molecule. So polysaccharides is this third category. The most important polysaccharide is cellulose. Cellulose is the most abundant organic compound on Earth. And that sort of makes sense. When you fly around in a plane, for instance, and look down onto the Earth, and you see a sea of green, all of those plants are composed primarily of cellulose. Cell walls are composed of cellulose. So cellulose is a polymer of glucose. It's just many, many glucose molecules bonded together. Usually maybe 3,000 glucose molecules or more bonded together make up a cellulose molecule. And those cellulose molecules are linked together by beta 1,4 bonds, or I think in the notes I call them beta 1,4 linkages. Those beta 1,4 linkages are those chemical bonds, those covalent bonds, is what gives cellulose its chemical characteristics. So I think the most important chemical characteristics of cellulose is that it's insoluble in water or organic solvents. So that's really important. Cellulose makes up cell walls. It makes up the structure of plant bodies. So it's important that it be insoluble. If it was soluble in water, obviously, then plants would just melt in the rain. So cellulose is insoluble in water or organic solvents, and it's quite resistant to chemical or enzymatic degradation or breakdown. What that means is that there are not many enzymes that can break these beta 1,4 bonds. What that means is that most organisms cannot digest cellulose or cannot digest it easily. In fact, when we think about organisms that do digest cellulose, it's usually actually microorganisms living in their guts that actually do the digestion of cellulose. So for instance, ruminants like cows or sheep that break down cellulose and derive sugars from cellulose, one, it's a lot of work. If you think about a ruminant that chews up grass or plant material and it goes down into their stomach, they usually have multiple stomachs. And there are microorganisms in their stomachs that help to degrade cellulose. So it's actually those microorganisms, the flora in their stomachs, that can break those beta 1,4 bonds. And even then it's difficult. So for instance, a cow, chews its cud. The material passes into its stomach and then comes back up and they chew it some more and it goes back down. So it's a lot of work to degrade or break down cellulose. Termites that derive energy from the sugars in cellulose, they actually can do that because of the microorganisms in their guts. So humans, we don't derive energy from cellulose. We pass cellulose all the way through our digestive tract. Now there's value in that. Obviously, when your doctor tells you you need more fiber in your diet, what that doctor is telling you is that you need more cellulose in your diet. And that bulk of the fiber moving through your digestive system obviously has benefits to us, but that cellulose ends up passing through our digestive system. We cannot physiologically access the sugars in cellulose for our metabolism because of those beta 1,4 bonds that we cannot break down. And the way cellulose is used to build cell walls, well, let's talk about that sort of building up the molecules. So here's a diagram of a cellulose molecule. It has, thousands more glucose going in each direction. That's a single cellulose molecule. And as we've discussed before, those cellulose molecules sort of stick together with hydrogen bonds. So here's an example of 3 cellulose molecules that are starting to coalesce together and are weakly bonded together with hydrogen bonds. And then they coalesce into microfibrils that might be, maybe in the neighborhood of 80 molecules of cellulose bonded together, and then those can bond together into macrofibrils. And those microfibrils and macrofibrils are what are actually laid down to form the cell walls. So cell walls are bundled together from these coalescing micro and macrofibrils that are built of individual cellulose molecules. So that's how cell walls are actually formed, is the cells lay down those macrofibril and microfibril bundles to form the cell wall material. And we talked before about how the angle at which those micro and macrofibrils are laid down ends up, for instance, determining the stiffness of the wood derived from those cell walls. Another really important polysaccharide in plants is starch. Starch is, in molecular form, is very similar to cellulose in that starch is made up of many glucose molecules bonded together. And in fact, the only chemical difference between starch and cellulose is in the nature of the chemical bonds that hold them together. Where cellulose had beta 1,4 bonds or linkages, starch molecules have glucose monosaccharides linked together by alpha 1,4 bonds. So just slightly different covalent bond holding them together. And in contrast to cellulose with beta 1,4 bonds, the alpha 1,4 bonds in starch are readily degraded by enzymes in many organisms. And that then makes starch useful for what it is, which is a storage molecule. Starch is the primary storage molecule for carbohydrates in plants. And so plants have enzymes that can easily break alpha 1,4 bonds that they need to. They need to be able to access the glucose molecules within starch and take glucose molecules and put them into starch and then take them out of starch again. So plants and other animals can break alpha 1,4 bonds and access the glucose. So we can metabolize starch. When we eat starchy foods, the enzymes in our digestive system are breaking down those starches, and we're accessing those glucose molecules in the starch for our cells to metabolize. So starch is useful as a storage molecule. And we talked about this when we talked about sink feedback inhibition. We talked about the buildup of starch grains made up of starch molecules in chloroplasts, remember? So we had a diagram showing the buildup of starch as starch grains in chloroplasts. The reality is that starch grains can accumulate in really almost any living cell in plants. So sugars can be stored not only in chloroplasts, but also in living cells, in wood, in branches, stem, or roots, for instance. So starch can be abundant in any tissue where there are living cells. So any living cells in plants can have starch in it where sugars are stored.